was the fact that there's been a lot of mixing between humans, what we call humans today, modern humans and these earlier groups. Evolution is supposed to move slowly, taking thousands of years to change a species. But when scientists looked at ancient human DNA, they found something shocking. Our genes changed way too fast, far quicker than nature allows. Brain power increased suddenly. Bodies adapted in the blink of an eye. What made us evolve so quickly? Could someone or something have altered us on purpose? Join us as we uncover the mystery hidden in our DNA. Evolution's speed limit. Evolution has always followed a simple rule. Change happens slowly. One generation passes traits to the next, and over time, small differences add up. A beak grows slightly longer. A limb becomes stronger. Fur changes color to match new surroundings. These shifts take thousands of years, sometimes tens of thousands, sometimes much longer. That is how nature works, and that is how it has always worked. The process begins with mutation, a random error that occurs when DNA copies itself during reproduction. Most mutations do nothing at all. Some cause harm, and a very small number provide an advantage. If that advantage helps an organism survive and reproduce, the mutation spreads. After many generations, it becomes common, and after many more, it becomes normal. This is natural selection, gradual and predictable, the foundation of everything scientists understand about the history of life on Earth. Scientists can measure how fast evolution moves by studying mutation rates in living species, comparing DNA across generations, and examining fossils to calculate how long physical changes take to emerge. The numbers are consistent, showing that major changes to body structure, brain size, or metabolic systems require enormous spans of time. A new trait does not simply appear but builds slowly, piece by piece, across countless generations. Human evolution should follow the same pattern. Early hominins walked upright millions of years ago. Their brains expanded over time. Tools became more complex. Language developed, and fire was controlled. Each milestone took vast stretches of time to unfold. From Australopithecus to Homo habilis, to Homo erectus, to Homo sapiens, the changes are visible in the fossil record, gradual and logical, fitting the model perfectly. But something strange appears when scientists look closely at the DNA of modern humans. The timeline does not always match, and certain changes show up too quickly. Traits that should take tens of thousands of years to develop appear in a fraction of that time, with entire sections of the genome shifting in ways that do not align with the slow grinding pace of natural selection. It is not just one gene or one trait, but multiple systems changing at once. Brain function, immune response, metabolism, language capacity, all transforming faster than the model predicts. This is not a small problem, but a fundamental contradiction. Evolution has a speed limit, and the genetic evidence suggests that limit was broken. The question is not whether these changes occurred because the DNA proves they did, but how. What could cause evolution to accelerate so dramatically? And what force could reshape the human genome in a time frame that should not be possible? Traditional explanations exist, including environmental pressures, climate shifts, and competition with other species. These factors can drive change, but they still require time and still follow the rules. What the DNA shows is something different, revealing change happening faster than those rules allow. Patterns that look deliberate rather than random, and a biological transformation that does not fit the standard model. This is where the mystery begins. Not with ancient ruins or forgotten legends, but with the code written inside every human cell. The code that reveals something happened to our species. Something that moved faster than evolution should move. Something that left a mark scientists are only now beginning to understand. And something that raises a question no one wants to ask out loud. If evolution has a speed limit and humans broke it, then what really changed us? Reading the ancient genome, for most of human history, the past remained locked away in bone and stone. Archaeologists could study skulls, measure jaw structures, and examine tool marks, but they could never see what truly made ancient humans different from modern ones. The genetic code, the actual blueprint of life, stayed hidden inside fossilized remains for thousands of years. Then in the late 20th century, scientists discovered they could read it. The breakthrough came from understanding that DNA does not always disappear when an organism dies. Under the right conditions, fragments survive in bones, teeth, and even soil. Cold, dry environments preserve genetic material best, which is why some of the most important discoveries come from caves in Europe and Siberia. When scientists extract ancient DNA, they're working with tiny damaged pieces, often degraded by time and contamination. It is painstaking work requiring extreme precision and advanced technology. Early attempts at ancient DNA analysis were limited and often unreliable. The first Neanderthal genome sequence, completed in the early 2000s, took years of effort and produced only partial results. But the technology improved rapidly, and by the 2000s, researchers could sequence entire genomes from samples tens of thousands of years old. What had once seemed impossible became routine, and the flood of data that followed changed everything scientists thought they knew about human history. The process itself is remarkable. A single tooth or bone fragment is cleaned and ground into powder, then treated with chemicals to extract any remaining DNA. The genetic material is copied millions of times using a technique called polymerase chain reaction, amplifying even the smallest traces. Sophisticated computer algorithms piece together the fragments, comparing them to modern human DNA, and filling in gaps where possible. The result is a genetic snapshot of someone who lived and died long before recorded history. What made this technology revolutionary was not just the ability to read ancient DNA, but the ability to compare it directly to modern genomes. Scientists could now see exactly which genes had changed when those changes occurred and how they spread through populations. They could track migrations, identify interbreeding between different human species, and pinpoint moments when the human genome underwent significant shifts. For the first time, evolution was not a theory built on fossils and guesswork, but a detailed record written in genetic code. The discoveries came quickly. Researchers found that modern humans interbred with Neanderthals, leaving traces of Neanderthal DNA in people of European and Asian descent. They discovered denizens with no complete skeleton ever found. They tracked how early humans adapted to new environments, developing lighter skin in northern climates, resistance to high altitudes in Tibet, and tolerance for dairy and pastoral societies. Each study revealed more about how humans evolved and spread across the planet. But the same technology that answered old questions also raised new ones. The more scientists sequenced ancient genomes, the more they noticed patterns that did not fit expectations. Changes that should have taken tens of thousands of years appeared in just a few thousand. Genes linked to brain development showed. Sudden shifts with no clear environmental cause. Entire sections of DNA looked as though they had been swapped out and replaced, not gradually altered through mutation and selection. The data was clear, consistent, and deeply puzzling. This is the foundation of the mystery. Not speculation or ancient legend, but hard evidence extracted from bone and analyzed in laboratories around the world. The technology exists. The data is real. And the timeline it reveals does not match the story evolution is supposed to tell. Something happened to the human genome, something fast and profound. And the only way scientists know about it is because they can now read the genetic record left behind. Chapter 3. Genetic changes that appear out of nowhere. When scientists first began comparing ancient and modern human DNA, they expected to find gradual shifts. Small mutations accumulating over time, traits spreading slowly through populations, the steady march of evolution doing what it always does. What they found instead were jumps, sudden changes that appeared without the expected transitions in between. Genes that looked one way in ancient samples looked completely different just a few thousand years later, with no clear path from one to the other. One of the most striking examples involves genes related to brain development. The human brain is what separates us from every other species, larger and more complex than it should be for a primate of our size. Scientists have identified specific genes that control brain growth, neural connections, and cognitive function. When they examined these genes in ancient DNA, they found changes occurring far faster than natural selection should allow. Variants linked to language processing, abstract thinking, and memory formation appeared suddenly in the genetic record, as though someone had flipped a switch. These were not minor adjustments, but fundamental rewiring of how the brain operates. The genes did not show the slow accumulation of mutations typical of evolutionary change. Instead, they showed abrupt shifts, with new versions appearing in populations within a time frame measured in thousands of years rather than tens of thousands. For context, it takes roughly 100,000 years for a single beneficial mutation to become widespread in a human population under normal conditions. Yet, multiple brain-related genes changed simultaneously in a fraction of that time. The immune system tells a similar story. Humans carry genes that determine how the body recognizes and fights disease, a critical factor in survival. Ancient DNA reveals that certain immune genes underwent rapid restructuring, with entire sections being replaced or modified in ways that do not resemble typical mutation patterns. Some of these changes align with known disease outbreaks, suggesting adaptation to new threats. But others have no clear environmental trigger and appear in populations that were not facing unusual health challenges. Metabolism is another area where the genetic record shows unexplained acceleration. Genes controlling how the body processes food, stores fat, and regulates energy shifted quickly in certain populations. The ability to digest lactose as an adult, for example, emerged in European populations roughly 7,000 years ago, far faster than standard evolutionary models predict. While this change is often credited to the adoption of dairy farming, the speed at which it spread remains difficult to explain through natural selection alone. What makes these changes even more puzzling is their precision. Evolution works through random mutation, a messy process that produces many failures for every success. Scientists expect to see genetic variation, dead ends, and leftover traces of failed adaptations. But in many cases, the shifts in human DNA look clean, as though specific genes were targeted and altered without the usual trial and error. The missing transitions, the lack of intermediate forms, the simultaneous changes across multiple systems all point to something unusual. Researchers have searched for explanations within conventional evolutionary theory. Population bottlenecks, where a small group survives a catastrophe and passes on limited genetic diversity, can create the appearance of sudden change. Selective sweeps, where a beneficial mutation spreads rapidly due to intense environmental pressure, can also accelerate evolution. But these mechanisms have limits, and they do not fully account for the patterns seen in human DNA. The changes are too fast, too coordinated, and too widespread. This is where the data becomes uncomfortable. The genetic evidence does not suggest evolution failed, but rather that something pushed it beyond its normal boundaries. The human genome bears the marks of rapid large-scale alteration, and the more scientists study it, the harder it becomes to explain through natural processes alone. The Neanderthal paradox. Neanderthals were not the brutish cave dwellers of popular imagination. They were sophisticated hunters, skilled toolmakers, and successful survivors who thrived across Europe and Asia for hundreds of thousands of years. When modern humans migrated out of Africa roughly 60,000 years ago, they encountered Neanderthals and interbred with them. The genetic evidence is undeniable, with most people of European and Asian descent carrying between 1% and 4% Neanderthal DNA. On the surface, this seems like a straightforward story of two species meeting and mixing. But the details reveal something far stranger. When scientists examined which Neanderthal genes survived in modern humans, they found a pattern that defies random interbreeding. Certain traits were kept, others were systematically removed, and the distribution looks anything but accidental. Neanderthal genes related to skin and hair, useful for adapting to cold northern climates, remain common in European populations. Genes affecting immune function, particularly those helping fight unfamiliar pathogens, also persisted and spread. These make evolutionary sense, beneficial traits that helped modern humans survive in new environments. But other Neanderthal genes disappeared almost entirely, and the speed of their removal raises questions. Genes affecting brain development and cognition, despite being present in early human Neanderthal hybrids, were filtered out of the population within a relatively short time. Studies show that regions of the genome linked to neural function are almost completely devoid of Neanderthal ancestry, even though those same regions should have been passed down through normal reproduction. Something actively worked against the survival of Neanderthal cognitive traits in the modern human genome. The same selective pattern appears in genes related to fertility and reproduction. Neanderthal variants affecting testicular function and sperm production are significantly underrepresented in modern humans, suggesting they caused reproductive problems in hybrids. This could explain why some Neanderthal DNA was lost, as natural selection eliminated traits that reduced fertility. However, the precision and speed of this filtering, combined with the retention of other traits, creates a picture more complex than simple hybrid incompatibility. What makes this paradox even more intriguing is the geographic variation. Neanderthal DNA is not evenly distributed across modern human populations. People of African descent carry almost none, which makes sense since Neanderthals live primarily in Europe and Asia. But among non-African populations, the amount and type of Neanderthal ancestry varies in ways that suggest selective retention rather than random inheritance. Certain populations kept more immune-related genes. Others retain different metabolic variants, and the distribution follows patterns that look intentional. Researchers have also found evidence that some Neanderthal genes were reintroduced into populations multiple times as those specific traits were being selected for and reinserted. This goes beyond normal interbreeding, where genetic mixing happens once, and genes either survive or disappear over generations. The pattern suggests active selection, with certain Neanderthal traits being preserved and propagated while others were excluded. The mechanisms for how this could happen naturally remain unclear. The timeline adds another layer of mystery. The filtering of Neanderthal DNA from modern human genomes occurred relatively quickly, within 10 to 20,000 years after interbreeding began. For evolution, this is an extremely short period, especially for changes affecting so many different genes across multiple systems. Natural selection can remove harmful traits over time, but the coordinated elimination of specific gene variants while preserving others requires a level of selective pressure that scientists struggle to explain. This is the Neanderthal paradox. Modern humans carry the genetic legacy of interbreeding, but that legacy has been carefully edited. Useful traits were kept, problematic ones were removed, and the process happened faster and more precisely than random evolution predicts. The question is not whether this filtering occurred because the DNA evidence proves it did, but how and why it happened with such apparent precision. Explanations that fall short. Mainstream science has never been short on explanations for rapid evolutionary change. When confronted with the puzzling patterns in human DNA, researchers turn to established theories, mechanisms that have driven adaptation throughout the history of life on Earth. Environmental pressure, climate shifts, competition for resources, and survival challenges have all shaped species over millions of years. The question is whether these forces can account for the speed and precision of changes seen in the human genome. Climate change is often cited as a major driver of human evolution. The planet has experienced dramatic temperature swings over the past 100,000 years, with ice ages alternating with warmer periods. These shifts forced human populations to adapt or die, creating intense selective pressure. Groups that could tolerate cold, find food in scarce conditions, or migrate to new territories survived while others did not. Over time, this pressure could favor certain genetic traits, spreading them through populations. The theory makes sense for some changes. Lighter skin in northern populations, for example, can be explained by reduced sunlight and the need to produce vitamin D more efficiently. Cold-adapted traits like changes in body fat distribution and metabolic rate align with survival in Ice Age Europe. Genes related to high-altitude adaptation in Tibetan populations clearly respond to environmental conditions. These are textbook examples of natural selection at work, and the genetic evidence supports them. But climate and environment do not explain everything. The changes to brain development genes, the restructuring of immune systems, and the rapid spread of cognitive traits do not correlate neatly with environmental shifts. There is no clear climate event that would drive simultaneous changes across so many different biological systems. More importantly, the timeline does not match. Environmental adaptation works over tens of thousands of years, not the compressed time frames seen in the genetic record. Even dramatic climate change requires time for mutations to arise, spread, and become fixed in a population. Survival pressure from disease offers another explanation. When populations encounter new pathogens, those with resistant genes survive and reproduce, passing immunity to the next generation. This mechanism has shaped human evolution significantly, with evidence of past plagues and epidemics written into the genome. The Black Death in medieval Europe, for instance, left genetic markers in descendants of survivors. But again, this explains isolated changes, not the coordinated shifts across multiple systems that characterize human genetic history. Competition with other human species has also been proposed as a driving force. When modern humans encountered Neanderthals and Denisovans, they competed for the same resources. Groups with cognitive advantages, better tools, or superior social organization would have thrived while others declined. This could theoretically accelerate selection for traits like intelligence, language, and cooperation. However, competition alone does not account for the precision of genetic changes or the selective retention of specific Neanderthal genes while others were eliminated. Population bottlenecks events, where a small group survives a catastrophe and repopulates, can create the appearance of rapid genetic change. When a population shrinks dramatically, genetic diversity decreases and certain traits become more common, simply because fewer individuals carry different variants. This phenomenon, known as genetic drift, can shift the genetic makeup of a population quickly. Yet bottlenecks also reduce overall diversity and create inbreeding problems, neither of which fully explains the patterns in human DNA. The most uncomfortable reality is that these explanations work individually but fail collectively. Each mechanism can account for specific traits or isolated changes, but none explain the broader pattern of rapid, coordinated, and precise alterations across the human genome. Natural selection requires time. Random mutation produces messy results, and environmental pressure does not target specific genes with surgical precision. The data shows something that looks less like gradual adaptation and more like intentional modification. And that is where conventional science reaches its limits. The unspoken possibility. There is a line in modern science that few researchers are willing to cross publicly. Careers are built on peer-reviewed evidence, reproducible experiments, and theories that fit within accepted frameworks. Suggesting that human evolution was guided, accelerated, or altered by something outside natural processes is professional suicide in most academic circles. Yet privately, in conversations away from journals and conferences, some scientists admit the data points toward conclusions that make them deeply uncomfortable. The resistance is not entirely unreasonable. Science has spent centuries fighting off supernatural explanations, insisting that natural phenomena have natural causes. The success of this approach is undeniable, producing medicine technology and an understanding of the universe that would have seemed like magic to earlier generations. To abandon naturalistic explanations, now especially for something as fundamental as human origins, feels like a retreat into the darkness of superstition and myth. But the genetic evidence does not care about professional comfort or historical precedent. The possibility that rarely gets discussed openly is that human DNA was deliberately altered not by gods or magic, but by a process that resembles genetic engineering more than natural selection. The precision of changes, the coordinated shifts across multiple systems, and the compressed timeline all point toward intervention rather than random mutation. The idea is not that evolution stopped, but that it was guided, accelerated, or supplemented by something with intention and capability. This concept is not purely modern speculation. Ancient cultures across the world told stories of humans being shaped, upgraded, or modified by beings from the sky or distant places. Sumerian texts describe the Anunnaki creating workers by altering existing creatures. Hindu scriptures speak of cycles where humanity is refined and recreated. Indigenous traditions from the Americas to Africa contain narratives of ancestors being taught, changed, or improved by external forces. Mainstream archaeology dismisses these as mythology symbolic explanations for natural processes that ancient people could not understand. But what if those stories preserved a memory of something real? The ethical implications of such a possibility are staggering. If humans were deliberately altered, then questions arise about autonomy, purpose, and identity. Were we modified to serve a function, to fulfill a role in some larger plan? Did the intervention benefit us, or did it come with costs buried in our biology? The idea challenges not just scientific orthodoxy, but fundamental assumptions about what it means to be human. It suggests we are not the product of random chance and survival, but the result of deliberate design. And that raises uncomfortable questions about who we are and why we exist. Research into these questions faces significant obstacles. Funding agencies are reluctant to support work that strays too far from conventional models, and scientists who pursue controversial theories risk marginalization. Data that does not fit accepted narratives is often shelved, explained away, or ignored rather than investigated further. Some researchers have noted gaps in the genetic record sections of DNA that appear incomplete or inaccessible, raising suspicions that not all information is available for study. Whether this is due to natural degradation, technical limitations, or something else remains unclear. There are also institutional pressures to avoid conclusions that might provoke public panic or undermine trust in science. If evidence emerged that human evolution was artificially influenced, the implications for religion, philosophy, and social order would be profound. Governments, academic institutions, and scientific bodies have strong incentives to maintain stability, and controversial findings are sometimes quietly suppressed or refrained to avoid disruption. This is not conspiracy, but caution, a reluctance to open doors that might be impossible to close. The unspoken possibility lingers in the background of genetic research, acknowledged in private, but rarely addressed in public. The data keeps accumulating, the patterns keep appearing, and the explanations keep falling short. Mainstream science insists that natural processes will eventually account for everything given enough time and research. But the timeline keeps tightening, the precision keeps increasing, and the question keeps growing louder. If the human genome shows signs of deliberate alteration, and if conventional evolution cannot fully explain what happened then, what force reshaped humanity, and why does acknowledging that possibility remain so dangerous? What the DNA still refuses to explain. The human genome has been fully sequenced. Scientists have mapped every gene, identified thousands of variants, and compared DNA across populations and time periods. The genetic code that makes a human is no longer a mystery in the technical sense. Every base pair, every chromosome, every segment of DNA has been cataloged and stored in databases accessible to researchers worldwide. Yet despite this completeness, there are parts of the genome that remain fundamentally unexplained. Sections where function and origin are still unclear. Roughly 8% of human DNA comes from ancient viral infections. Fragments of genetic material inserted by retroviruses millions of years ago. Most of this is considered junk, inactive remnants with no current purpose. But some of these sequences play active roles in development and immunity. Their original viral function repurposed for human biology. Scientists do not fully understand how or why certain viral DNA became integrated and functional, while other sections remain dormant. The presence of this foreign genetic material raises questions about how much of the human genome is truly human, and how much was borrowed, inserted, or acquired through processes still not completely understood. There are also long stretches of non-coding DNA regions that do not produce proteins but appear to regulate gene expression and control biological processes. For years, this was dismissed as evolutionary baggage, leftover sequences with no real function Recent research has shown that much of it is active and important, influencing everything from brain development to disease resistance. But the mechanisms are complex and not fully mapped. Scientists know these regions matter, but they do not always know what they do or how they came to exist in their current form. Certain genes in the human genome have no clear evolutionary predecessors. They appear uniquely human, with no obvious relatives in other primates or earlier hominins. Some of these genes are involved in brain function, language, and abstract reasoning, the very traits that define modern humanity. Their sudden appearance in the genetic record without the expected transitional forms remains one of the unsolved puzzles of human evolution. Standard explanations involve rapid mutation and positive selection, but the lack of intermediate stages makes the story incomplete. Then there are the gaps in ancient DNA itself. Not every population has been sampled. Not every time period has been sequenced, and not every individual left recoverable genetic material. The picture scientists have assembled is based on fragment snapshots from specific locations and eras. There could be entire chapters of human genetic history missing, events that occurred in regions where DNA does not preserve well, or among populations that left no trace. The conclusions drawn from available data are only as complete as the data itself, and the data is far from comprehensive. Some researchers have noted anomalies in the structure of certain genes patterns that do not fit typical mutation processes. These are not errors or contamination, but genuine oddities, sequences that look as though they were inserted rather than evolved. The scientific community has been cautious about interpreting these findings, preferring to wait for more data rather than draw premature conclusions. But the anomalies persist, and with each new study, the list of unexplained genetic features grows rather than shrinks. The most unsettling conclusion is that the human genome, despite being sequenced and studied extensively, still holds secrets. There are questions it raises, but does not answer, patterns it reveals but does not explain, and gaps it contains but does not fill. Evolution provides a framework for understanding human origins, but the framework has holes. Places where the data does not fit neatly and where alternative explanations become harder to dismiss. If humans were altered, once the evidence would be written in DNA. And if that evidence exists, but remains unexplained, misunderstood, or inaccessible, then the most important question is not what happened in the past, but whether it could happen again. The genome is a record, but it is also a blueprint. And blueprints can be read, copied, and modified. The code that makes us human is no longer locked away in bone and stone. It is open, readable, and vulnerable. And that is perhaps the most unnerving revelation of all. If you enjoyed this video, like and subscribe, and also click the next video shown on your